Well, first of all, let me apologize for that long title. I think it's the longer one for this conference, so just forget about it. And well, in this presentation, I will try to introduce to you the topic of dynamic combinatorial chemistry, and for this, I will use an example to show you how these systems can be used. Then, first of all, and as an introduction, I would like to talk to you about the dynamic combinatorial chemistry and also about the halophilic proteins, which in principle are quite different topics. Then, the first one, the dynamic combinatorial chemistry, what proposes is the creation of a library of compounds interconnected through reversible chemical processes. What this basically means is that from a set of small chemical entities, and by joining them in different ways or different combinations, we can generate a large chemical diversity or a large number of new chemical entities. Then, since these bonds in this union are actually reversible bonds, we've got a thermodynamically controlled process, and so at the end we've got that the concentration of each of the generated compounds in the mixtures depend on its relative stability. Then, well, these kind of systems can be studied from different point of views, one of them being the systems chemistry, which proposes to study the emerging properties of these kind of systems, or to check or to see the systems as a whole. Then in the other side, we have the halophilic proteins, which are those proteins of the halophilic archaea, which are a kind of bacteria that live in highly saline environments. These bacteria, well, what they do in order to adapt to this high salt concentration is to increase at their surface the number of glutamic and aspartic acid residues, also to decrease the overall hydrophobic content, and to decrease the solvent accessible surface. So overall in this introduction, we've got a set of evolutionary trends observed in nature, and also a system which is able to adapt depending on some external stimuli. So our purpose for the next experiment was that we could use one of these dynamic combinatorial chemistry libraries and to study it as a systems chemistry approximation, let's say, in order to parallel with some of the evolutionary trends observed for the halophilic proteins. So what we did was to synthesize a set of building blocks with this general shape containing amino acids and so protein-like information, and also containing thiol groups which can oxidize forming disulfides, which are reversible bonds. Then the very simple experiment we did was to mix 3 of these building blocks, which were the ones based on glutamic acid, also the one based on glutamine, and the one based on serine. And what we observed when mixing these building blocks in equal concentrations was that they were able, in the mixture, to generate dimers, the 6 possible dimers, also small amounts of trimers. Actually, we could detect the 10 possible trimers, sorry. Nothing very interesting so far, but then what we did was to increase the salt concentration. And very interestingly, what we observed was that some of the concentrations of the dimers actually changed. Would it represent the amplification factor of these dimers in front of the salt concentration? And we interestingly observed the amplification pattern predicted for the amplification or the single amplification of the dimer gathering together 2 units of glutamic acid. Moreover, if we take into account now the trimers, we did observe the amplification of those trimers, gathering together 2 or even 3 units of glutamic acids. So overall, we've got a system which is actually able to parallel with the evolutionary trends observed for the halophilic proteins. Well, I hope this presentation has been useful for you to see how dynamic combinatorial chemistry can be used for the experiment modeling for the evolutionary trends. Thank you.